and then began at the University of Alberta as a CRC chair tier 2 in 2002. Okay, so Sally has a great history of producing excellent research and service on many different levels. She has been chair of the SICB Division of Evolution and Developmental Biology, has been president of the Canadian Society of Zoologists, board of directors of the Canadian Scientific Submersible Facility, council member for Ocean Networks Canada. And then broadly speaking, Sally works on the evolution of coordination mechanisms, marine and freshwater, largely focusing on sponges as one of the earliest branching metazoan groups, and one that lacks a nervous system, which for me isn't really a good thing, but that's okay. And then the work that Sally will talk about comes from her last sabbatical leave, which was 2018 to 2019 in the United Kingdom, where she did work describing the genome of freshwater sponges. Okay, Sally, so I'll let you take it away.
Thanks, Declan. So I'm going to try sharing here. Whoever's host has to enable my screen sharing.
Yeah, and it's not me anymore.
Sorry, Dan, I did it. You should be co-host now, Sally. No?
Nope.
How about now?
Yep, now we go. Okay. You know what? This is I don't know about you got everyone else here, but this is the story of our teaching. It's sort of a familiar story every week, just to ensure. And hopefully the internet works, because on Tuesday I was teaching here and the internet was kind of like interesting. And so I then taught in my office, and I'm like dodging between them. So let me know if I freeze up. Someone give me a shout, speak, because it's hard to watch the little chats. Okay, so I take you can see my screen. Someone, Declan, give me a thumbs up. Is that good?
Yes, I can see it.
Okay,
Yep.
good. Okay, the right screen, correct? Just the sharing screen. Okay, so thank you, Tracy, for the invitation last year on arm twisting, and I put this off as long as possible. It was a busy year last year for me, for NSERC and all kinds of things. So I just ended up having 2 lectures this winter. It's not because I've been jumping up and down, believe me. And so because I have spoken before about the ecophysiology work we do in the lab today, I decided to tell you about genome work we do, which is a little bit different. This is work that we published in 2020, came out during COVID, when we were all kicked out into funny places. So it was put together with a very collegial group of people who I've been working with for some time, who came together and completely selflessly threw themselves into this project. The principal folk who made it happen are Nathan Kenny, who was postdoc at the Natural History Museum in London, Anna Riesgo, who was there, is a colleague of mine from a long time, Joe Ryan, who's at the Whitney Lab in Florida, and Warren Francis, who was postdoc at the Southern University in Denmark at the time. And then other colleagues, April Hill, who I work with closely, she's in Maine, and Gert Verheide, with whom I started this project in 2010. And as I said, it was published, much to our great pleasure, in Nature Communications in 2020. So what is it? Why sequence the genome? So obviously we've learned a lot about relationships among metazoans and all kinds of organisms from molecular-based phylogenies. And many of these relationships have been sorted out, but there are some still very contentious parts, and they're shown here in this tree on the right with the dotted lines. In particular, the area that I work with is the base of this metazoan bush, and I'm interested in aspects of animal evolution of animal characters And to do this, we need to know a lot about the main suspects. The usual suspects really in this group are of non-bilaterians are sponges, placozoans, cnidarians, shown here by a little anemone, and ctenophores, which are gelatinous sea animals. And then bilaterians are not even depicted with a creature. So in this sort of set of relationships, there are some there are a number of sort of principal questions that remain. And I'm picking out four. First is the question of cell types, the evolution of cell types, a big one for people today. Then is, in fact, whether things like placozoans actually have lost an extracellular matrix. They're an enigmatic little group of less than a millimeter-sized creatures that are marine. Could cnidarians have lost bilateral symmetry? So that's another big question. And then the real elephant in the room is this one I put last, which is whether things like ctenophores either evolved came about before sponges, or whether they actually came about later, in which case have they evolved a nervous system convergently. If they came about first, have sponges lost a nervous system, or do we have two different, many different times that nervous systems have come about? So in order to really answer these questions, you have to have more information on all of these groups. And the big problem is that we haven't had enough model systems with which to be able to do this. So this has really been highlighted by recent work, like this paper that came out in November, that got a lot of press about, it was a gene expression single cell study of a freshwater sponge called Spongilla. And a large group of authors claimed a number of things, but including the fact that there was a cell that they called choanoneuroid. And they suggest that this neuroid cell connects itself to the feeding structure of the sponge. And hence these news articles say more than a gut reaction and so on. And so the problem here is that those of us who were interviewed about this work were all rather cool, because it's not the first time that someone has claimed that there might be neurons in a sponge. This has come about many, many times in history. But very difficultly, the data are really quite circumstantial, and we're not really sure how correct they are. It's difficult to repeat them because we don't have this data, and we're not all working on this sponge. So we need, in fact, a lot more baseline data on more sponges, and we need more people working on even the same sponges in order to make some headway and really understand this. So we need more accessible species. So that might be a claim that everyone makes, but in sponges, we really only have one genome. I call it the bread and butter genome. It is Amphimedon queenslandica, and many of you use the sequences in comparative analyses you might do. It's found here on the Great Barrier Reef. This is Heron Island. It's the field station of the University of Queensland. And I actually discovered this little sponge in my postdoc work, lucky place to work, when I was there as a postdoc in 1998. So its real asset is that it produces broods of embryos, and these swim out into little larvae, and you can collect hordes of them in the shallow, in the seawater that's calm. And so because of these and other attributes, it was decided to sequence its genome. That was JGI that sequenced it. And that was published in 2010. And it really has become the benchmark genome. So what we've learned from it is quite a lot. We know that it has about 40,000 genes. This is quite good coverage. There are some things missing. For example, only 3 Winks there. There's various little bits and pieces that seem to be absent. We know about its signaling. We know it has a photopigment that's cryptochrome. It responds to light. We have evidence that GATA might have a shared role in phagocytosis and something to do with the endothelium eating. It has a very compact genome. It is sort of considered an intermediary between non-metazoan and metazoan genomes. We also know that it's highly methylated. So we know quite a lot of things from this. And the group that can work on this has done a huge amount of work to make our general understanding really good. The biggest problem is it lives in Australia. Nice to go there, but difficult, and no other group can actually work on it. So why do we not have other models? There are some, and I put a whole suite out here in a paper in 2015. There are a number that different people work on around the world, but only one of these, the freshwater ones, are available throughout the world, and they're starred here as they're found in all of these different places. So the freshwater sponges, a lot of the people who work on marine things think they're a little boring. It's very interesting how that comes about. But I discovered that they were very practical to work on when I received this video from my colleague, Yoko Watanabe, in the late 90s. This was actually just when I was starting my postdoc in Australia, and I discovered that you could hatch them in the lab. They would grow up. This is a time-lapse film from the Tokyo Film Corporation, and it shows probably every 20 seconds an image. And so they grow up in a dish within a couple of days to form a little filtering sponge. And not only that, if they are given ink or dirt to filter, they filter it out and it makes them convulse. And they do these concerted contractions that then effectively expel all of this junk out of their filtration system. And it was this coordinated set of convulsions that convinced me that this was a very practical group to work on. So I began to work with them in the lab and discovered you could collect them in the winter as these little tiny balls that you find on logs or on the rock. And they will hatch out into a little sponge. You see here from its fried egg stage. So it's cyst in the middle and it's tissue around the edge and the filtering chimney on the right. And so we call this a gemule, and these are its canals. And so you can grow armies of these from individuals, which are clones. And so each individual is either a male or female, and you can sort of hatch out hundreds to thousands of one clone, or you can use different clones for diversity, especially if we don't know if there's differences in behavior between the different individuals. They are have practical tools, so we can do in situ, we can do RNAi, and in addition to that, amazingly, they can be frozen at -80, and you can hatch them out after years and years and years. And I have shipped these all over the world now, and they have grown them in 3 different continents. So from ones that I have shipped. So it's a very practical model. It is also found in beautiful places. So these are lakes on Vancouver Island. It has this pan-continental distribution. So really in a Northern Hemisphere contribution. It likes to live where areas that there are a little more nutrients, although not dirty water. So interestingly enough, here's an article from 2000 from Chicago wastewater treatment plant that showed that once they had got the water so clean, they were checking this with divers, and they found that the walls were coated with sponges. So this sponge, and freshwater sponge in particular, but Ephydatia, is known to colonize these kinds of systems, drinking water systems. And in fact, the clones that I used for the genome come from the drinking water system, the head tank of Victoria. And so this is a map of Victoria and the Souk Reservoir up here, and there's a 8-kilometer long tunnel that connects that drinking water system to Colwood, where it's treated. And that head tank is inspected every 2 to 3 years for integrity. And I was invited by the people at the CRD to come along with them several times, and this is what the head tank looks like. And in fact, its walls are completely coated with sponges. So this is what the sponges look like in the dark of that head tank, and in the winter, where they form these tiny little cysts, which you can almost see as little balls there on the surface. And in the head tank, there are about 3 species that live on the walls. There are a number of other things that live in there, like you can find some bryozoans. I found fish. There are a few other freshwater invertebrates, but by and large, it's sponge. So one of the things about it is that getting sponge is one thing. Making good DNA from sponge was another. And I collected a lot of sponge, I made a lot of DNA, and then we discovered that even though I started this project in 2010, shipping that DNA didn't work. It degraded somehow. We started to wonder how that worked. We tried all kinds of things, and it just basically became a very long project that wasn't going anywhere. It's very frustrating for a lot of people. And so in 2017, I learned from a colleague at Harvard that she had sequenced the genome of an A seal using Dovetail Genomics. And so I contacted them and discovered that they would promise a chromosome level assembly. Now, it wasn't going to be cheap, but it was cheaper than using it for ongoing salaries for lots of people everywhere. And so we made this decision to go ahead with Dovetail. And so Dovetail's data is shown here. They used PacBio sequencing. They made Chicago libraries, and then they made 3 Hi-C libraries, Dovetail Hi-C libraries using Illumina. And they did scaffolding with HiRise. It's their software. And they now have quite a reputation for doing this in a very fine way. What they had is an input assembly and then their HiRise assembly that they did with the assembly. So they did the entire thing. In fact, it took me several months to go back and forth with them to get the DNA to be of the quality to go directly through into sequencing, because that seems to be the hitch. And so they went from having an N50 of the lengths of 92 scaffolds of a megabase to having only 11 of a 9 megabase scaffold size. So a really impressive assembly. What does that look like? So that can be visualized here in this essentially dot plot of the read and the mate position. And what you see on this is the resulting 24 main scaffolds, which are actually plotted here on a bar graph and correspond remarkably to the chromosomes of the sponge. And fortunately, we have a karyotype of Ephydatia and of a number of different freshwater sponges, which have been done by a group in Japan, by Shijima, and a remarkable postdoc who did this. We contacted them because we tried to do more karyotypes, and it turned out we were unable. Several of us in our team were unable, and he claimed that nobody in their group was able to make such quality karyotypes. Again, so it is a very challenging thing, but you can see nevertheless, there are 2 large chromosomes here and then 22 small ones. And these correspond pretty well to all these scaffolds. We have 24 large scaffolds, but in fact, one of our scaffolds, scaffold 24 and 7, turn out to be split in 2, as was recently confirmed by a paper that just came out yesterday. So that's really neat that it captured all of these different chromosomes. And the other one, the 25th one, turned out to be the full genome of a single microbe. And one of the things about this genome is it is a very clean genome. It hasn't got any other microbes except that one, which is really unusual because pretty well every Ephydatia that you will collect has many microbes and lots of algae things. But this I managed to culture up xenically, and it came out of the head tank, so it was in the dark. So it is possibly one of the reasons why it's also a good assembly. So this is probably the most impressive first view I got of the assembly. It was put together by Warren Francis, who was the postdoc in Denmark. So what it shows is the size of the circle here is the N50, which is essentially that the size of more than half of the genome sequence contigs. So 50% of them are greater than this size. So this shows that this is this 9.8 megabase size of those contigs. It's a very large contigs. Only 2 other species that we could find to compare with had equivalent size goods assemblies. So Trichoplax adherens is a placozoan, shown over here, and then Branchiostoma floridae is a lancelet. And there's a new assembly now of Branchiostoma, but this was the one we could use. So what you see on the y-axis is the number of scaffolds. So you see that Ephydatia has about 1,400 scaffolds and total. So 24 of the genome of the chromosomes, and then lots of little tiny ones that probably need to fit in somewhere. And so you have many fewer scaffolds in Branchiostoma. And then down on the x-axis is the genome size. So Ephydatia is about 326 megabase genome. Branchiostoma bigger, and Trichoplax very small. But what you see from this is that there's three sponges here, Amphimeda, Tethia, and Sycon, and they all have lots of scaffolds. They're very, very fragmented. Even Nemeopsis, which is the ctenophore, is also very fragmented. It's still not got a really contiguous assembly. The other Nigerian we've been doing a lot of model work with is Nematostella. It's also very fragmented. And even Hydra, although Hydra has a new assembly that again has just come out this week. So, but this is pretty nice view of the genome. So one of the things you can do with a chromosomal level assembly is it allows us kinds of comparisons that you can't do with a fragmented one. And one of them is to look at macrosynteny. And so synteny is basically the retention of gene linkages on the chromosome. And so synteny sort of on the ribbon is what it means. So we compared the position of the genes from Ephydatia to Trichoplax, and this dot block can be read by the number of green dots show the homologous gene pairs across these two. And so you see little dense spots of them there. Those show that shows that there's patches of macrosynteny. And if you look at two scaffolds here, this is scaffold 22 from Ephydatia, and this is scaffold 5 from Trichoplax. Each of these shows, each black line shows a homologous gene, and the green lines show its match to the homologous gene in another species. So all the way down here or all the way up here. And then you see even Ephydatia against the lancelet, Branchiostoma, there's lots of patches of macrosynteny, and the same shown here as genes crossing to their homologous gene pair. And so what we found, though, is that while you get this linkages conserved across to chordates, you get a lot of shuffling of order within a chromosome. And that seems to be, that's a typical story. So, but you can actually see what's happening here. And to see that that is not happening to non-metazoans, we compared Ephydatia's genome to Monocyga and Salcadica, which are two quantiflagellates, and we see no synteny showing there. But between the two quantiflagellates themselves, we see those patches showing up again. And then also between two placozoan species that are sequenced, you see really tight sets of synteny, suggesting these are less divergent. So that's one type of analysis that can be done. Another that can be done with the kind of sequencing we had from Dovetail, the high C sequencing, is it allows study of gene regulation. And so what this does is it is a method that it cross-links DNA regions that are really spatially close in the nucleus. And then you can compare what the kinds of regulation that might be going on between within the DNA, within the chromatin at these points. And so in mouse, for example, you see these points here that are called topologically associated domains. They show up as squares, and on the tips of them, they have loops, which are sort of regions that are keeping this set of DNA together. So this is from another paper. And then on the right, it shows the scaffold number 1 of Ephydatia compared to scaffold number 1 here. And it shows, this is analysis done by Ramon Rivera, who's a PhD student in Gert Verheid's group. And he used the Bowtie and Homer in order to come up with this plot. And it shows the tags and loops, but they're not actually connected. And in fact, we're not really sure. And this kind of analysis probably has new softwares that can be and approaches that can be done to it now. But Ephydatia doesn't have these CTCF sites that makes the loops, so it would be unclear how that would work at all. So definitely this data lends itself to more analysis of that sort. So one member of our group was Alex de Mendoza, and he had previously studied DNA methylation as a means of gene regulation across non-bilaterians. So here a sponge, Amphimedon, another sponge, Sycon, and then the Ctenophore Neopsis and the anemone Nematostella. And one of their main discoveries in that work, which came out in 2019, so really just before our paper, so he was very keen about this, was that the sponges had a very highly methylated genome, which was actually comparable to vertebrates. And the idea there is that it allowed a lot of innovation in gene regulation in vertebrates. And so what's going on in sponges, they weren't sure, because the other non-bilaterians they studied did not have that. So he was keen to analyze the Ephydatia DNA, and I shipped him off some to Australia where they did this, and they discovered that Ephydatia had an intermediate level of methylated genomes. So it would suggest that not all demosponges and certainly not all sponges have such a highly methylated genome. And so this was sort of an interesting thing. And what it tells us is that we need more sponge genomes. I will say that even today, I had an email from Sanger sequencing group who are trying to sequence 50 sponge genomes along with a massive symbiosis project. And it's not so simple. They're having a lot of trouble with the assemblies, even of really good quality material produced. So I have 4 species in there in that sequencing system. So coming up with more genomes for this is just not, it's not a matter of just throwing it at it. It's a lot of tricks to actually get them to this level. So what do you get from that? We see that it's very useful to see across the board. This is the genome size of Ephydatia, the freshwater sponge. It's almost twice the size of Amphimedon. So Amphimedon is a very compact genome. It's similar to this Cnidarian, and you know, it's not that large genome as they go. This is just showing the assembly, so it has a low number of scaffolds. But importantly, sponges do confirm they come out to have about twice the number, or far more genes than most animals, and twice the number of vertebrates or humans. So that's a very interesting thing. There's 39 to 40,000 genes in Ephydatia and Amphimedon, and even in Sycon, not shown on this particular plot. And it's not got anything to do with repetitive DNA. These seems to be very similar across the sponges. There's differences in intron size, gene size, and so on among the different sponges, but the number of genes is actually coming out to be the same. So I guess one of the things that we asked then is, like, why is that, and what does the genome tell us about the number of genes? So freshwater comes with its challenges, and freshwater sponges made that big transition about 311 million years ago. This is a plot that comes from a previous analysis based on calculated on fossil data, so it's calibrated. And they made a divergence, so they diverged into their different groups only 10 to 30 million years ago. And actually, that's really interesting. Why did they diverge? And I think it might have something to do with when migration of birds came around, because how did the sponges get into fresh water all around the world? And one of the ideas is that they get attached to the feathers as birds go foraging, and they do get transported perhaps in fish guts, but probably more so in birds. So that's a really interesting question that nobody has followed up. So they live in all these different habitats, and they even live in trees. They live in really cool places around the world. So this is a picture from a PhD student in Brazil who took a picture of a freshwater sponge in the Amazon. And here, when the water is filled up in the rainy season, they are completely submerged in water. And as the water drains in the dry season, they dry out and they live on these trees. And they even live in deserts there where they get swamped and then they dry out and they get swamped and so on. So they live in a huge range of environments. And so that comes with a lot of different challenges. And so what we did is we looked at the gene gains and losses that might have occurred in these different groups. And this work was done by Ana Riesgo. And so this compares the gene gains and losses under two different scenarios, where the sponges are considered the sister to other metazoans or where the ctenophores, because there's always this idea that maybe sponges lost a lot of genes, or maybe that's because their sister, the ctenophores came about first and sponges have lost everything. But the analysis shows that in fact here on the bar graph, these are the gene gains. So in fact, sponges have gained a lot of genes. In either scenario, the gene gains are about 12,000 more gains since their divergence from the other taxa. So they've really gained a lot of genes compared to other groups. And if you compare the losses, which are these large circles, so in this case, in the sponge sister hypothesis, the sponges have lost about 375 genes. And in the ctenophore sister hypothesis, they've lost about 471. But in both of those scenarios, the ctenophores have lost a lot more. So the reason for the vast number of genes appears to be that there's been a lot of gene gain without the similar loss. So they've retained them. So I guess what are some of these gene gains? What are they doing? We looked at a few of them very cursorily, I must admit. This is analysis that Warren Francis did. And so here you see there's huge duplications in integrins, for example, and in GABA receptors. And you can see those if you go to the JBrowse, our genome browser. You can see duplicated. So these genes appear many times along the scaffolds. So that's a very interesting way of studying it. If we look at the GABA receptors, so here he's plotted an alignment of views showing they have these transmembrane receptors. They come out as regular GABA receptors. 120 are predicted just here on scaffold 22. And you can see in his gene tree here, his freshwater dimmer sponges have their own set of metabotropic GABA receptors here. Then these are the glutamate receptors. And then you see other dimmer sponge ones, and the hexactinoids, which are the glass sponges, have a separate grouping. And so Warren wasn't really sure, but maybe they're involved in some aspect of being in freshwater. So he was suggesting chemosensation and so on. So this is an area ripe to be discovered, to be explored. Another thing we did together with Ana Riesgo is I studied the differential gene expression over a number of different stages of development, because you can grow them in the lab. They hatch out to these very, very stereotypical stages. And so I grew them up and harvested 3 different individuals at these different time points. And then we looked at the genes that were expressed. And what you can see is that they express different genes in the early stage before they've hatched. And those are sort of pictures depicted in scanning EM on the right. Before they've actually clambered out. They go through a period of getting ready to hatch. And so what's going on in that stage before the cells have come out to form the body is different than what's going on in these other early stages of development. And then what's going on there, which is about skeletogenesis, various types of adhesion and so on, what is being expressed. And in this latter stage, which is the fully formed functioning filtering sponge with its little vent and its responsiveness, this is a different set of genes. And these you see metabolism, homeostasis, epithelial genes, and so on. I mean, the only other really interesting thing here in this, this is a rich set of data that people can use and are using frequently, but you can see here that sponge-specific genes are turned on after it hatches, which is quite interesting. So that's another thing to be analyzed. So something to do with forming this body plan is a sponge-specific process. And then we have in blue Ephydatia-specific genes, and then in purple the eukaryotic-specific genes. So we did a lot of different analyses of the types of pathways that were there. And April Hill looked, for example, at genes in the Wnt pathway, and in green you see all the little boxes of all the Wnt pathway genes that are present in Ephydatia, and you see it's quite populated. So pretty well everything you look for tends to be there. And we know a lot about the Wnt pathway in the sponge. It's an area that she and my students have been working on together. It seems to be involved in forming the body plan and the polarity of the filtration system. Another thing that that April is quite interested in, and she and I are now exploring together with Shun Sagaris, a postdoc in our group, is the use of Ephydatia as a model for symbiosis. And so here is a little egg, fried egg. So this is the Ephydatia living with no algae, and you can grow them up azealically in the dark, and like the ones I get from the head tank, completely free of algae, and you can rid them of algae, and then you can reintroduce symbionts. And then you can see those here sitting in amongst flagellated chambers, which are shown in green here in a cross section of confocal. And you can see that in the sponge, here's a quanocyte, which is its feeding cell. There are cells in the mesohyl that have both algae and bacterial microbes. So it's a very interesting system. These are intracellular symbionts. So there's a lot of work that can be done with this animal, but I'm going to wrap it up here to say that voila, we have a high quality genome, which we're pretty pleased by. People are still asking for information on how to make good DNA and do this. It has really long contigs. We have chromosomes. We can see that there's synteny across to lancelets even. There's a lot of data there that can be used to explore DNA regulation, which is actually being used currently. There's a lot of information to be used here for understanding this freshwater transition. And in particular, I didn't go into aspects of sensory systems, channels that are involved. There's a huge number of transient receptor potential channels that we've been studying that are involved possibly in sensing the water flow through them. And just, it really becomes an excellent model for other people to use because you can grow it in your lab. And so what we've also done is made available a website, which I was very fortunate to have assistance from the faculty put this up on our website, where you can get all of the resources. So in here, you can find the genomes, the models, the JBrowse link to explore the genome with all of the different data. There's a blast server and also the differential gene expression data. And we pretty well get about 90 hits, visits a month. So people do use this, and that's what the point was, to make this a much more accessible thing. And even last night, this paper came out, which, as you can see, has used Ephydatia. So Ephydatia is here as the sponge comparison for understanding in greater depth the level of synteny and actually the evolution of chromosomes. It's a really cool paper. If you are interested, it literally came across my feed last night. So I'm pleased to say that it is actually of use, and I think this kind of quality is worth it for people to be able to do a lot of different work in the future. So that work comes with money, and there's a lot of different people pitched in on this, but in particular, I really, like everyone here, I know we are so grateful for NSERC for giving us the ability to make these steps in different directions and support students and other people to be able to do this work. And then I also want to say what's not ever acknowledged on here is the sort of Debbie in the storeroom who shipped things for me so many times, and the various people across campus who've made that possible. Because without all of that assistance, without people being able to do that, receiving and delivery and also running all these grants, we couldn't do it. So I really want to put a pitch in and thank all of the staff for helping us with that. And that, I said I would thank you for listening, for coming this afternoon, and I'm happy to try to answer any questions.
Thank you very much, Sally. That was fantastic. I'm sure there'll be a lot of questions, a lot of hand clapping going on. So thanks for those reactions. And if you'd like, in terms of questions, can you please raise your hand electronically via the reactions, and then we'll call on you in order as those questions come up. Thanks so much, Sally.
I'm gonna let you handle it.
Okay, great. Heather has a question.
Hi, Sally. That was fantastic. Some of it went right over my head, like as was already answered. What the heck is a scaffold? You know, I hear the word and then I look at it and I go, I don't think it's a ladder or anything like that. But what I'm really just glommed onto is that cool idea of the diversification of freshwater sponges being tied to migration of birds. You know, obviously that is how they get around for the most part to otherwise unreachable areas. And so there'd have presumably something similar might have gone on with freshwater bryozoans. Do you know if anybody has done a time calibrated phylogeny that includes freshwater bryozoans?
I don't know that. I know who I could ask though, who's at the Natural History Museum, who might know that. They might be very closely tied together, considering they like to live together.
Yeah. So who did you find in the bowels of Victoria's water system? Did you get your bryozoans identified?
No, I don't think so.
Okay.
I think I have one. Bring it
Because there
over
would be
for you.
that's another model system for you to start working on. You know,
It's
when
true.
you get
It's
bored
true.
of sponges.
In my spare time. I
Yeah,
keep saying that. Exactly.
Thank you.
Great, thank you. Tracy.
Great talk. Thanks, Sally. I was just wondering if you see any signatures of horizontal gene transfer from microbes in any of your genomes.
We do.
Cool.
There's one we comment on. I think it's in the aquaporins actually, but there are examples. There are several, and you have to go in and start picking them apart. But yes, they look like it. People have different opinions on it, but we think there are. I can send you those examples if you're interested.
Yeah, that's fascinating. Very cool. Do they correlate at all with the microbes that you know are present with the sponges? Or
so this particular sponge, remember, had none, which is crazy. I'm trying to think. Those particular genes, I don't know. I have, again, this is one of the wonderful things about this talk. It's very diverse on things, so I wouldn't know that specific. But I'm betting you that it does. And I think you'd have to look at other sponge genomes that aren't so clean as this to actually see that the ones that we're seeing in the sponge are the ones. In the sponge are the ones from the microbes in the sponge.
cool.
Right.
Thanks.
Great, thanks so much. Looks like John is next.
Hey, great talk, Sally. Just going back to your introductory slide. So with your genomic analysis, do you have now any clues about the possible parallel evolution of nervous systems?
Yeah, so parallel, yeah, so convergence, it would, so it's completely an opinion-based thing, John. If I go back to those ones that I even showed you of the one from Detlef Arendt's group, so if you analyze the genes carefully, like in our paper, we studied snares, for example, a PhD student from Joel Dagg's groups on our team. And he looked at snare proteins, which are involved in synapses maybe, and in vesicle trafficking broadly. And those show that they are essentially non-neuronal snares. They've diverged independently, and so you would say that these are present as a family, but they've not a neuronal type. But if you have the opinion, and if you don't go in depth into the phylogenetics of it, a lot of people will say that they're all there and that you've actually got a homologous system, right? You've got homologous proteins, but we don't find evidence for that. So it very much looks like that sponges never had anything and never lost anything. And that doesn't really help us on the story as to convergence of the ctenophore nervous system. It just says that we don't see evidence for them in this group. So now we need to know where ctenophores fit in the picture of things, and that story is really going to be problematic, right? So I don't know if that explains it very clearly, but basically so far the data suggests that yeah, there could be convergence, but we don't see evidence of any of nervous system characters in this sponge or in sponges.
Neat. Thank you. Sheila's next.
Hi there. That was a really great story, Sally. It's really fun for me to see that. I have two questions. One is, do you find transposons in your genomes? And the second one is whether do you find any evidence for developmental changes in methylation that would suggest epigenetic gene regulation?
I'll go for the second one. No. Is that okay? No, we didn't do developmental stages for DNA methylation. He just did one sample, so we didn't have the different stages. So that needs to be done. That would be good. We didn't do the transposon. I can't remember on the transposons. So you're gonna have to. I'll have to get back to you on that. Yeah, sorry.
That's okay.
Yeah.
Thank you.
Nice
Great,
to
thanks,
see you.
Sally. Oh, sorry, Sheila, did you have another question or?
No, I'm good.
Okay, great. So Kathy has a question, and then there is one in the chat I'd like to get to after Kathy.
Sally, that was really cool. Your transcriptome showed, I couldn't miss it, immune response genes turned on at about your third stage. And I wonder if that's about the same time that they get colonized with bacteria or.
That's really interesting. Yeah, there could be things going on in there, because even in a dish in the lab, you're going to get bacteria in there anywhere, right? We're not growing them in a laminar flow hood or anything like that, for sure. So that's definitely something to consider. I suppose one could challenge them even more and do it at different stages in order to determine that to kind of push it in one direction. Yeah, and those genes are ready to be examined.
I'd love to see a list of them. I think it would be fun. I recently in my lecture talked about that the immune system is, perhaps the immune system is actually there to keep commensals in control rather than defend against pathogens.
Right. There's some good discussion on that, isn't there? I've read that too in the sponge microbial literature. Yeah.
Exactly. Yes.
Yeah.
Very cool. Thanks.
Thank you.
Great, thanks, Kathy. So Sally, the question in the chat is, and this is from John Bruner. So you showed in your cladogram that the freshwater sponges radiated at the beginning of the Cambrian. Is it known what the salinity of the oceans were at that time, which was less than what it is now?
I don't know. Beginning of the Cambrian too. How do we? I don't know that it was less. I guess it's changed over time. The timing of the first sort of idea of when freshwater sponges or the group might have come about is a little, of course, you know, it has a big range, right? We put in 311, that's from a Schuster paper from Gert Verheid's lab. So they didn't diversify till much later, which could have been, you know, transporting around, I guess. But I don't know. I honestly don't know. I would love to know more about that, what the salinity of the oceans would be. And I do know somebody else who could tell me about that, probably Eric Sperling, who does a lot of these geological analyses. So I'll make a little list of these little chat questions and follow up on them because they're great. Thank you. Sorry, I can't answer you better.
We are coming to the end. If I can probably steal maybe the last question before we take you off the hook here, Sally. When you looked at sort of some of the gene diversification or duplication events, you talked about GABA receptors, and all of the receptors that you had there for both GABA and glutamate appeared to be metabotropic type of receptors. Mm-hmm. Why specifically metabotropic, or these sort of G protein, GPCRs? Is there anything specific to that?
Yeah.
Any reason why?
Yeah, in this sponge and in a number of the different lineages, we only see metabotropic receptors. We don't see ionotropic receptors, but there are some groups. The homoscleromorphs have them. They're in Calcarea, and they're in distinct demosponge lineages, but they're not in this. No ionotropic receptors. So I don't know. So they're lost. That is certainly something that's been lost, and that may be freshwater or it may be lineage specific.
Okay,
yeah, it's kind of frustrating that it's frustrating they're not there. I would like them to be there for other reasons or for signaling, but I think the question as to why they would be lost and why they're in some lineages and not others is very interesting.
Cool, really neat. Well, that was a fantastic talk, Sally. I mean, a lot of really neat stuff and so interesting. And I'm glad you took the time and effort to do this and to share that with us. So again, thank you on behalf of everybody. Thank you very much for an excellent talk this afternoon.
Well, thank you guys for all listening. I appreciate it. Have a good weekend, everyone.
Yeah, thank you. You too.
Early weekend, yeah.
Tracy, and thanks so much for help with this.
No problem.